Hello! Welcome to Squid Essays. Inking your academic pen. My name is Julie and I will be your instructor as we read through multiple biochemical activities. The article source is embedded in the video description below. Feel free to read along. Please note that you can get a custom, plagiarism-free essay for as little as $10 a page at Squid Essays. The link is in the description. Mutation is the change in the DNA sequence of cell genome which are heritable. It causes various changes in the gene number and the chromosomal number of the genome which causes to the mutagen. Mutagen cause manipulations in the genetic material during cell division either by exposing to ultraviolet ionizing radiation, mutagenic chemicals, or viruses. Mutations are the heritable changes in the genome. Some mutations may cause changes in the phenotypic characteristics of the gene expression. In microbial organisms they are designated as wild-type strains and agents that cause mutations are called as mutants. Therefore, mutants are characterized by the inherited differences between them and their ancestral wild-type strains. Variant forms of a specific genetic determinant are called alleles. Genotypic symbols are lowercase, italicized specify individual genes with a plus superscript indicating the wild-type allele, whereas phenotypic symbols are capitalized and not italicized. For example, to produce SS-galactosidase, in order to ferment lactose, the genotypic symbol used is LacZ+, and mutants not producing SS-galactosidase are LacZ. The lactose fermenting phenotype is designated as Lac+, and inability to ferment lactose is Lac. Classification on basis of selection. Mutation are classified as selectable and unselectable. A selectable mutation is that mutation in which there is a growth and survivability under a specific environment conditions where the mutant can grow and wild type will die. An unselectable mutant requires a cell-by-cell examination in order to find a mutant with desirable characteristics. Selection can be direct or indirect. The indirect selection is used for isolating mutant that is deficient in producing a necessary growth factor. Like E. coli can grow on glucose and mineral salts more than lactic acid. Related research articles, multiple biochemical activities of NM23/NDP kinase and gene regulation, Department of Molecular Biology, Princeton University, Princeton, New Jersey, 08546-5414 NM23/NDPK protein play an important role in the cancer in the development. These proteins play a role in the DNA metabolism including binding of the nucleotide and the transcription and the cleavage of the DNA strands using covalent DNA complexes. DNA cleavage reaction of NM23/NDPK is characteristics of DNA repair enzymes. It is seen that both DNA cleavage and the NDPK reactions are conserved between E. coli and T human beings. NM23/NDP kinases are regulators of gene expression during development and the cancer via unrecognized pathways and mechanism of genetic control and unrecognized role in DNA repair and recombination. The Parkinson disease-causing LRRK2 mutation I2020T is associated with increased kinase activity. Christian Johannes Glockner, Norbert Kinkel, Annette Schumacher, Rolf J. Brown, Eric O'Neill, Thomas Meidinger, Walter Kolch, Holger Prukish and Maria Suffing. Parkinson disease is the most common neurodegenerative disorder. Recent studies have shown that mutations in the leucine repeat kinase 2 gene, located on the chromosomal region, is the cause for the disorder named as Parkinson disease. The LRRK2 protein consists of multiple domains, contains a predicted kinase domain, with homologic-OMAP kinases. Using cell fractionation and immunofluorescence microscopy, visualize that LRRK2 is located in the cytoplasm, which is associated with cellular membrane structures. LRRK2 protein shows the autokinase activity. The disease-associated I2020T mutant shows a significant increase in the autophosphorylated eosinophil type protein in vitro. This suggests that the PD is caused by the I2020T mutation, which is associated with an increase rather than a loss in LRRK2 kinase activity. Mediator protein mutations that selectively abolish activated transcription, the deletion of any one of 3 subunits of yeast mediator as MED2, PGD1, HRS1, and SYN4, of transcription regulation terminated the activation by Gal4 VP16. Other mediators' functions like the stimulation of basal transcription and of TFIIH kinase activity will remain unaffected. The overlapping mediator subunit was found for activation by GCN4. So, the genetic requirements for activation in vitro were closely related with the same in vivo. A whole genome expression of A9AYTZ2Y strain showed diminished transcription of a subset of inducible genes but that show only minor effects on basal transcription. These findings make an important connection between transcriptional activation in vitro and in vivo and identify mediator as a global transcriptional coactivator. DNA gyrase and topoisomerase 4, biochemical activities, physiological roles during chromosome replication, and drug sensitivities. DNA gyrase and topoisomerase 4 are two topoisomerases present in the bacteria. They play a crucial role in the cell. Topoisomerase gyrase is involved in the chain elongation during the replication of the chromosome, whereas topoisomerase 4 separates the linked daughter chromosomes during the terminal stage of DNA replication. The different roles can be attributed to the various biochemical properties of the two enzymes. Therefore, biochemical activities, physiological roles, and drug sensitivities of the enzymes are reviewed and studied using gyrase in the topoisomerase. Natural selection and the emergence of a mutation phenotype, an update of the evolutionary synthesis considering mechanisms that affect genome variation. In evolution, it is assumed that the mutations are random with respect to their potential effects on survivability. The phenotypic variations that affect the survivability of the descendants, intrinsic variations and the location of genetic changes under natural selection. Through the action of natural selection on the genes, favorable mutations can become more probable than random. From the example, the variation in bacterial surface proteins to the vertebrate immune response, it is seen that a genetic change is better than random with respect to its potential effect on survival. However, some potentially mutations are so probable that they can be viewed as being encoded in the genome. An updated evolutionary theory includes emergent under selective pressure of genomic information that affects the probability of different types of mutation that results into the genomic survival. References: http://www.springerlink.com/ content /p45443688510474m/ http://hmg.oxfordjournals.org/ CGI/ content/ full/ 15/ 2/ 223 http://www.pnis.org/ content/ 96/ 1/ 67. http://rjournals.annualreviews.org/ doi/ abs/ /anurev.microdoc.0908555. Thank you for taking your time to listen to me. I hope you enjoyed learning about multiple biochemical activities as much as I did. Kindly remember that you can get a custom, plagiarism-free essay at Squid Essays for as little as $10. The link is in the description. See you next time.